Welcome to this probability and statistics session. Roberto Oliveira, the chair today. In the interest of time, I'm not going to make long introductions to our speakers, so without further ado, Jonathan Taylor.
Thank you, Roberto, and thank you for the invitation to present a talk here. So I'm going to talk a little bit about selective inference, as my title suggests, which is, hopefully I'll define that a little more clearly as we go along. So just to start, as a caveat, this is a sort of embarrassingly untechnical talk, and I did that partly on purpose because the audience, I presume consisting mostly of mathematicians, might not be familiar with many words a statistician might use, so there won't be too many overwhelming technical details. And I'm going to start off with a sort of motivation for this line of work that is not just myself involved in, but many statisticians, and it's the replicability crisis in science. So it's not hard to, if you look through scientific journals, general science journals these days, it's not hard to find articles like these ones that express worry in the scientific community about reproducibility of results and or replicability of results, those two terms being slightly different. Or here's another article, statisticians issue a warning over the misuse of p-values, that is, misuse of statistical methods is a worrying field, worrying scientists these days. Okay, so that's the general motivation for this talk, and I'm going to talk about a few sort of solutions. And I'll just talk, if there's any sort of common mathematical theme in what I'm going to talk about, it's the sort of, imagine having a convex set K, and it's the normal cycle of K, and that is K here being this cube. It's the set of all points in the cube combined with all their normal vectors. And this will come up in a few different parts of the talk as, as an object that we use to resolve some of these problems, or in my resolution of some of these problems. Okay, so as I said, it's not hard to find in the general literature of science concern about replicability and reproducibility. And it's also not hard to find in the statistics literature. So a few years ago, Emmanuel Candes, in his Wald lecture, he made the point that today we're at a point where scientists collect data first and ask questions later, and that this requires the need for statistical methods that can live in this sort of new world of science. On the other hand, the scientific method has sort of long assumed that scientists use data to generate questions, right? This is a sort of classical iterative process of science. And Tukey was a big proponent of exploratory data analysis, and he said things such as, the idea of a scientist struck as if by lightning with a question is far from the truth, meaning that scientists really do need to use data to generate questions, though they often have to confirm them as well. So there's sort of two broad themes that are kind of in conflict, and this is the conflict that I think Emmanuel was talking about. Exploratory data analysis, and this is something that all of us can do freely now with much freely available statistical software. Take some data, evaluate some functions on it, maybe visualize some plots, look through the data, comb through the data to look for interesting patterns. And scientists have really always done this. They always did it on data they had at hand, and they formed new questions, which they might then later design a new experiment to confirm. So what is confirmatory data analysis? This is the sort of part of statistics that is really the core inferential part of statistics. And this is, the standards of science require some sort of confirmation of your discovery, and often this is statistical. And so this is the place where p-values and confidence intervals and other such things enter the picture. And there is this conflict between these two types of data analysis in that it's misleading to naively use the same data for both exploration and confirmation. And I'm going to start off with a sort of simple example demonstrating this that's going to stick with this for a while. So I want to now sort of think about how a scientist interacts with data in their everyday life. And I want to I'm going to represent this by a sort of dependency graph in the computer. And we're going to sort of start thinking about doing statistics in the context of a dependency graph like this. Okay, so here is maybe a typical data scientist or scientist use workflow. They might have some data t, and throughout the talk, t and t prime will be data. And they have the data in the computer, so they're going to evaluate some functions. They're going to look at some functions of t, and I'll call those queries. So he's going to evaluate some function q1 first. U is for user. That maybe is another node I could put in this graph, but I've sort of left it off for the time being. And then after observing that, well, we'll do some more exploration. We'll call another function, evaluate another query. The query we choose is going to depend on what the user saw in the first stage because he's interacting with this data, and it's also going to depend on the data. And then maybe based on this, they're going to draw some confirmatory sample T prime, for which they might try and publish a paper. So more concretely, the first query might be choice of a tuning parameter. There are many statistical methods out there that need some tuning. The second query might be, I'll choose some important features for whatever this statistical task is. And if you're not familiar with exactly what I mean by these two terms, I have a simpler example that I'm going to stick with coming up shortly. Of course, this is a very simple example. There could be more data collected. Science is sort of an iterative process, so we might run more and more experiments and get more t prime, t double prime, etc. And of course, I only evaluated 2 queries here. I'm going to, in principle, be looking at more than just 2 aspects of the data. So an example we'll come to again at the end of the talk is suppose T is some data that consists of measurements on HIV viruses that measures specific mutations in the virus and how these viruses respond to a drug in vitro. So that's a kind of typical problem we would call a supervised problem in statistics. And we might want to build a model to explain the in vitro resistance as a function of mutations. So we'll typically have some model selection step where we're going to first find some important, what's called the main effects. And then maybe having chosen the main effects, we'll look for some interactions. And then maybe we'll draw confirmatory data, or maybe we won't. This T prime could just be the sort of empty node, in that this function just returns null. There's no new data coming. Okay, so that's a slightly more realistic example. And here's a simpler example that I'm going to stick with for longer now. So this is an example in the clinical trial literature called the drop the losers design. So this node T here, it denotes we have k different drugs or k different dosages of a drug, and we're trying to measure what the best dosage is for a certain population. So you can think of we have T different samples from with the same population treated with k different drugs or different dosages of a drug. And the first question we ask is which drug has the best outcome? So that's the query 1 we're going to ask. So that's going to be a function of the sample we drew in the first case. And maybe, the second query, let's also ask, just for a slight variant, let's ask what was the second best drug? That's not always asked in drop the losers, but I just had 2 queries here, so I had to give a second one. And this data T prime here is based on which is the best drug. We're going to collect a new sample, or the best dosage, a new sample at that dosage, and try and estimate, the treatment effect. Okay, and the gold standard here would be we'll just take the confirmatory sample and, report the average of the treatment effect in the confirmatory sample. Okay, well, if you're running a clinical trial, you spent a lot of money to collect all this data to make the decision about which is the best drug. So it might seem kind of wasteful to only use the data in the first stage for selection, and you might want to reuse the earlier data. And so this is the sort of first example of this conflict between exploratory analysis and confirmatory analysis. Deciding which drug or which dosage is the best, you've done some exploration of the data, and you want to report an estimate, so a sort of confirmatory aspect. And mixing these 2 stages can lead to problems. So here's a simple example. So I have 2 sort of estimators of the treatment effect, and I'm plotting here sort of the distribution of the kinds of errors they make. So the gold standard is this curve over here, and its errors are centered around 0, so it's an unbiased estimate of the treatment effect. It's wider than this red curve. The red curve, what is it? This is the unadjusted estimator. And I've sort of set this problem up to have equal sample sizes in the first stage and second stage. There are, of course, variants. They don't have to be equal sample sizes, etc. Okay, the unadjusted estimator is biased because, and why is that? Because of the winner's curse. In the first sample, TK was chosen because it was the largest of several of them. And this is a sort of a simple manifestation of this sort of conflict about collecting data first and asking questions later. It's also called researcher degrees of freedom in the psychology literature sometimes, and also called the winner's curse. And I just want to emphasize that this is a very simple version of data analysis. If you're going to have really complicated queries here in dependency graphs, it's rather easy to lead yourself to bias. And so the gold standard is always unbiased here. It's a little more variable than this one, but this one is biased. And I could exaggerate this difference very easily if I imagine I had many different drugs and they all had roughly no treatment effect. Then this red curve would sort of shift to the right. I could change the width of this histogram by changing the number of samples in each group, etc. Okay, the main point of this is that naively using the unadjusted estimator is going to be biased. And this will be also true if you try and test the hypothesis that there is no treatment effect, or if you try and construct a confidence interval for the treatment effect. Okay, so not all of us might know what those two terms mean, but it's easy to see that this is a biased estimator. Okay, so now I want to talk about some ways we might remedy this bias in such a way that we can reuse some of the data in the first stage. And that's, we want to sort of partially resolve this replicability crisis. So in the literature, there are two terms, reproducible research and the replicability of research. And these have slightly different definitions. There's been a great amount of work in let's say, the computational science in order to make it easy for you to document this dependency graph in such a way that if we give someone else the data t and t prime, and we've documented what these functions are, we're going to get the same answer. So that's an important part of this program. But at the same time, that doesn't really address any of the statistical problems that can come about by slightly more complex data analysis. And I would say to address replicability, we really need to statistically understand this sort of collection of random variables. And what do I mean? How are these a collection of random variables? Well, if this is random, then these two being functions of t are of course random variables, and t prime is just a confirmatory study. So this is really a collection of random variables, and we sort of want to understand statistically how these behave. Okay, so now here's a sort of rough definition of selective inference. And I want to say, for today, I'm going to assume this is valid inference in the presence of possible selection effects based on these queries Q1 and Q2, right? And or whatever queries you might have looked at. So let's see, let's take a step back and think about how and if this was an issue in the classical scientific method. And I've sort of taken my dependency graph and I've added a bit of colors to it, and the colors I'll explain as we go. So in what follows, this is yellow, but for this slide I sort of made it a faint yellow to denote that in the classical method, we sort of lose focus of data we collected in the past, and for inference we only really care about the confirmatory sample. So I focused on this blue note here. And I see part of my slide has dropped off the bottom. That might happen once again, but okay. So the exploration for the user is limited to the exploratory sample t, and there's no interaction with the confirmatory sample, though this earlier data is wasted because we've forgotten about it by the time you get to the confirmatory stage. And there's no selection effect because once we collect this data, the question of interest has been fixed. That is, if I'm talking about estimating a quantity, I know what I want to estimate, or if I want to test a hypothesis, I know what the hypothesis is. But we've sort of forgotten all about this, so we've sort of wasted this data. Okay, and now what I want to do is try and understand, not waste this data, and sort of understand a little bit better about how it behaves. And let's think of some of the challenges we might face in that. So one of the challenges is that if we're allowing these queries to be, a scientist interacting with this data, their question or the queries they evaluate, they don't always translate nicely into statistical objects. And what do I mean by a statistical object? Well, these are things coming from the language of statistics, right? In order to do statistics properly, at least inferential statistics, we need a statistical model. And in our gold standard example, that is a model for the distribution of the confirmatory sample given actually the exploratory sample. So that's some collection of distributions. There has to be some parameter of interest. That's a function on the statistical model. And there are, we have specific language in statistics about parameters, talking about a point estimate or confidence intervals, or even if we were Bayesian's, a posterior distribution. And if these are sort of arbitrary queries, it's not really clear that when the scientist sees the result of these queries, that it's going to immediately cleanly translate into something that fits into this framework here, that is a statistical model with specific parameters. And there is something, I know there is something cut off on this slide. I know what it is, so I'll say it. So I would say, Tukey said scientists are not struck by questions as if by lightning, but I would say that if scientists are struck by anything, they're not struck with null hypotheses, or they're not struck with confidence intervals. They're not struck with that. They don't necessarily think this way. Okay, but there are some sort of prototypical data structures that do sort of fit it that do naturally lend themselves to a statistical model and a set of parameters. And this is what has been called large-scale inference. Here is a simple example, and I'm not going to try and describe it in any great detail. But we measure a disease status D for all subjects in a population, and some genomic signature, some genomic information at each position along the chromosome. Here we have data from the 23 chromosomes, and we measure something like some association between disease and that particular marker status. So if I have 10 million positions along the chromosome, I have 10 million different associations I might be interested in. Each association is tied to a particular location in the genome. So there's a natural choice of parameters here. So if I specify a model for the data, theta I would be the association for marker I with disease D. And down here I've written this data is T prime, a sort of confirmatory sample where I measure disease status and the marker at each location. Okay, so this is sort of work in the general area of selective inference that has been ongoing for many years, and that is because this sort of problem fits into this notion of multiple comparisons, having many different parameters to estimate. These being parameters, there is a well-defined statistical model. I can talk about distributions, so these are estimates of the parameters. I can talk about distributions of the estimates of the parameters, and we can sort of fit it naturally into the statistics language. And so there's many approaches in multiple comparisons. One that I don't have time to talk about, but that has been hugely influential in statistics, is this notion of making a decision about each of these, say, 10 million hypotheses that controls something called the false discovery rate. And in the interest of time, I cannot go too much more into this. Another approach in the literature is this idea of controlling the family-wise error rate. And these are, roughly speaking, the statistical problem there becomes understanding the distribution of the largest of a collection of normalized association measures. These being z-scores, the maximum of a collection of z-statistics. And I'm just going to take a small aside here to bring back that picture of the normal cycle. So other work that was going on in parallel to the FDR work, and maybe started a little bit earlier, considered thinking of these markers as being measured from some continuum and sort of modeling a continuum of the distribution of the maximum of a large set of z-statistics. And there's various approximations out there. I'm not going to define these too precisely, but there is an old sort of approach based on the volume of tubes formula to approximate this probability. And I won't say much more about that than to say that this is the first sort of example where this normal cycle comes up. So if I want to compute the volume of a tube, will it become some integral over the normal cycle around the base set m? And here's an example of the tube around the cube. To compute the volume, you must add up all of these different pieces. And, well, I won't dwell on it. OK. So now let me sort of pause here to go back to the original goal here. And remember, I wanted to sort of understand how these collection of random variables behave in such a way that meshes with the way scientists use data today. So does this, I want to sort of allow, use workflow to sort of inform this or to be part of the statistical model. And I would say that these large-scale inference problems I described, they're really basically just confirmatory analysis. There's a sample, I have a set of hypotheses I'm interested in, and I'm going to try and control false discovery rate or control family-wise error rate for models of that confirmatory sample. So you can use simultaneous inference in some sense to allow interaction with the data in terms of these queries. That is, you can construct simultaneous confidence intervals based on the Bonferroni or volume of tubes methods that could be used. So you could say that by using simultaneous inference that allows for all possible choices in these first two queries, you could get an answer that covers the true parameter with high probability. So that's one way to say that maybe these large-scale inference methods can be used here. So an argument against this is really, once I've told you that I'm going to measure these 10 million diseases associated with these 10 million genetic markers, really those 10 million questions are already determined, right? I didn't have to look at the data in order to decide I wanted to ask those 10 million questions. So, OK. So now I want something that's going to allow the scientist to look at the data and decide what questions are interesting before running a confirmatory analysis. So now here's my graph again with the nodes, and I've not faded them out. I've not faded the yellow out in this picture. And remember, in sort of classical inference, we throw away everything in the first stage. And we just consider the distribution of the confirmatory sample given everything we know so far. And so formally, throwing away this exploratory data is really the same as conditioning on it. And then just specifying a model for the distribution of T prime given everything we've seen before. OK. So the yellow nodes are nodes for which I'm going to condition. T prime, the blue nodes, are nodes for which the user or the statistician has to postulate a model. So has to say, this is what I believe the distribution of these blue nodes is. And OK, this is unfortunate that the formula dropped. Maybe I can see if I can make it. So there is if I just do this, that might be better. This formula might be somewhat useful. OK, so in the drop the losers design, what's the difference? Well, we don't throw away all the first stage data. We have a model for the first stage and the second stage data jointly. And so we just condition on what we've seen about the data, that is, which was the best and which was the second best. And we sort of do inference in this model for the first stage and the second stage data. And it's not hard to see that you can get a better estimator than the gold standard by Rao-Blackwell. That is, taking the unbiased estimator, which is the one valued on the confirmatory sample, and conditioning it on sufficient statistics of what we've seen so far, as well as or conditioning on the fact that the best treatment was the kth one. And so this is Rao-Blackwell applied to a model for the data, conditioned on what we've seen about the data. And that kind of model is what I'll call a selective model. OK, and if this is officially to be Rao-Blackwell, I guess I'm making the assumption that it's normal and I know the variances, but let's grant me that for today. OK, and so you can see there is a better estimator. So we would say statistically that the gold standard is sort of inadmissible in this case. That is, they're a better estimator. This blue one, that in this example is only slightly less variable than the gold standard, but is also unbiased. The unadjusted one is still biased. That is the same. I'm showing this for the estimator, but you can do similar things for testing hypotheses and confidence intervals. And here I just condition on the first query, but we can construct similar intervals conditioning on the second query as well. Okay, and so here's an example. I changed the data generating parameters a little bit. In this case, you can see that we really do better than the gold standard. So in this case, this Rao-Blackwell estimator, the blue curve is less narrow than the gold standard. And actually, you can't really see, but it sort of coincides almost exactly with the unadjusted estimate. Now, so if for this data generating mechanism, I had just used the unadjusted estimate, it would turn out it would be roughly unbiased. It would be a pretty good estimate. Of course, I don't always know a priori whether the unadjusted one is going to be good or not. And this conditional one sort of interpolates between regions where the unadjusted one is bad and where the unadjusted one is good. And Rao-Blackwell says that the blue one is always going to be better than the gold standard. Okay, so that's the sort of name of the game we're going to play, and we're going to try and do that in more generality. Okay, so quickly, so the classical scientific method, it really does allow you to intervene. Really, what the scientist just has to make a model for this data once he's seen everything about the exploratory data. And what he's doing then is really specifying a model for the law of confirmatory given exploratory. So the general approach, I'm just going to specify a model for the data, the whole data itself, but I will condition on what I've seen about the data. And we're going to do statistics on all of the data, that is, construct hypothesis tests, construct confidence intervals, and point estimators based on all of the data, but I will take into account what I know about the data by conditioning. So now, is this improvement I showed you, is this limited to drop the losers? No. Do I really need a confirmatory sample? Not really. Actually, we could even work in the case where t is the same as t prime. And can I allow the scientist to do arbitrary questions? Probably not. So let me see what kind of things we can do. Okay, so what do I mean when I said do statistics in this model? So that means I'm just going to do statistical inference in a new model that I'll call the selective model. And the scientist has to posit a model for what generated the data. And I'm going to adjust, call that model M. I'm going to adjust it to be the model M star by just adjusting each distribution by conditioning it on what we've seen about the data. And this function zeta star, which is up to proportionality, the likelihood ratio between each f star and each f, that in theory you can read off this graph. Once I specify a distribution over here, well, in principle I can work out this function zeta star. Okay, so if I want to carry out this program, the theoretical crux becomes, well, transforming what we know about statistical models M to these models M star. There's a lot known in mathematical statistics about consistency, central limit theorems, and if we posit a model for which we know something, like central limit theorem, what does that say about the model M star, and can we use that? So that's that requires some mathematics, and one can try and answer that question. And in a computational sense, I really need to get my hands on this function zeta star, and that can be rather complicated for many queries. Okay, so let's see how I'm doing on time. I think I have about 10 minutes left, or perhaps 15, but so I think I'm 10 plus 5 minutes per question, hopefully. Okay, I'm known for never finishing my slides, so but I think I might today. Okay, so let's take a step back and look at what we did for drop the losers. So in drop the losers, the first query, what are we doing? We're looking for the best treatment arm, the best treatment dosage. And you can write that as maximizing over the simplex, a linear function of alpha and the treatment in the first stage. And so I'm going to make a new variable that is t prime minus t, and there's a small bit of sleight of hand here in that I'm assuming in this description here that we collected data in the second stage for all the arms. I don't really need that, but it just sort of makes this description a little bit easier. So if I had collected data for all the arms in the second stage, I would have, I could define omega to be the difference of the confirmatory data and the exploratory data. And then this maximization is the same as maximizing the same linear function over the simplex. Well, alpha with let's say the full data estimator, t plus t prime, over 2 if I wanted to get the actual estimator, but that doesn't matter for maximizing, minus alpha dot omega, where omega is this t prime minus t. And so this is just a perturbed version of maximizing on the full data, which is the best treatment. Okay. And so now you could consider, what does it mean to solve this maximization problem? We get something about, there are the first order conditions of solving this optimization problem. Say something about the solutions, alpha star, in terms of the input data, which in this case is t, which I can write as t plus t prime minus omega. And so you can take the first order conditions for solving that optimization problem. And they can be restated as saying that omega is equal to the full data plus or minus a cone. And that cone is, well, it's the normal cone of the simplex at the optimal point. And here I'm assuming I have a density, so there's an almost surely unique optimal point, but let's not dwell on that too much. So I can write this function, zeta star, that I have to modify my distributions by as an integral of, well, something involving this g omega, this is going to be the density of the randomization omega, integrated over all possible omegas that would give me the same optimal value, having fixed t plus t prime. Okay, so this says if I want to use this in a computer, if I have any data, any distribution for t plus t prime, I can find this, this function zeta star implicitly by sampling from omegas in this set with this density here. So if I sample them jointly in the computer, I will get the desired distribution for my full data t plus t prime. Okay, and if we could only do this in the drop the losers design, this would be kind of boring because it's just a simple clinical trial example, but you could do this in greater generality. So I'm going to define call this a randomized convex program. So maybe I have some loss, squared error loss, logistic regression loss, or some convex loss in beta. I'll put a penalty. There are lots of penalties in favor in statistics, structure reducing penalties, and I'm going to randomize it by perturbing it the same way drop the losers did, by just adding this linear term in beta, and I'm going to generate the data omega myself on the computer. Okay, and what kind of penalties might I have? Well, there are a lot of penalties like the L1 norm that induce structure on the solutions that people like to use to find simple models or simple estimators. Okay, and let's consider, oops, something is missing my subgradient equation here. Let's consider solving this, this optimization problem. I can write that as the gradient being equal to 0, or more properly, the subgradient being equal to 0. And I'm missing the term omega in this subgradient equation, but pardon me for that. So it should really be that u hat, the subgradient of the penalty at our solution beta hat, is omega minus the gradient of our loss at beta hat and data. And I'm going to allow the scientist to ask questions as follows. I'm going to allow them to ask questions about beta hat and u hat. And this is really what drop the losers was. Right? Drop the losers actually asked us to identify u hat. What was the maximizer of that convex program? Okay, and here's this picture again, the normal cycle. Where does this come from? Well, in studying the optimality conditions, there's this condition that u is in the subdifferential of the penalty at beta. And for many of these norm penalties, structure-inducing penalties, this is the same as saying that beta is in the normal cone of the k, which is the I guess, the dual unit ball of the penalty at the point U. So there's a reason this picture showed up again. And this is actually, this being the cube, this you could think of as the dual unit ball of the L1 norm. And you can see here that, it's easy to see here that the L1 norm gives sparse solutions by looking, say, at this part here. This is, say, this face of the 2 cube. And if my subgradient is in this face, then the normal cone is just a vector, is generated by a vector pointing out in this direction. So whenever I have a U in this face here, my beta, my solution, beta hat, is going to be 1 sparse with a positive sign in, I guess, let's call this the first direction. Okay. So now how can we work with this? Well, we can compute this adjustment factor, zeta star. What is it going to be? I'm going to integrate over all U and beta that give me, if Q is my query and I got the answer little q, I have to find all U and beta that satisfy that. And I'll integrate this density. This is a sort of change of variables Jacobian, and integrate it against appropriate surface measure for the normal cycle. And I didn't put a dimension here for the surface measure just because, well, sometimes the dimension might change. If I actually show you the whole subgradient, like it's kind of ambiguous what this dimension is, but I don't want to get hung up on that. Okay, so what's the payoff of doing this? Well, there are lots of penalties out there in statistics, and discovering structure that is like sparse, useful structure in data can often be cast as solving a convex problem and looking at the solution. So there are quite a few queries that can be fit into this. A canonical example being the lasso, which I'll call the noisy version of compressed sensing. So if I put the L1 norm here and I have some convex loss, the solution is going to be sparse for large values of lambda. This, if any of you have paid any attention to statistics in the last 20 years, has also, of course, been hugely successful. So, and this picture actually shows what we have to do for the lasso, this being actually the normal cycle of the L infinity norm in R3. Okay, so now suppose my query is I'm going to show you, I'm going to solve a randomized version of lasso. So I have my loss plus L1 penalty, and I perturb it this way. I'll perturb it with a little bit of a quadratic term just to make sure I have a solution if the number of features is bigger than the number of samples. And I'm going to show the user the actual whole subgradient when I solve this problem. And we'll call that the observed, that's the query that will allow people to use. And then this adjustment factor has a specific form. It's an integral over one particular normal cone in that normal cycle. That normal cone is determined by the actual observed subgradient. So it's like an integral over a piece like this sort of 2-dimensional cone, this 3-dimensional cone, this 1-dimensional cone, or I suppose sort of 0-dimensional cone if I had the dense solution. Okay, and of course, if I tell you can ask a convex question of the data and look at the query, you can build up quite a few reasonable workflows with this. You can combine more queries, more data nodes, and we still have a well-defined notion of what it is we have to do to adjust our model. So here, if this was my model for the used data analysis, the adjustment factor can be expressed in this particular way. I will have to know what each of these queries are, what these convex functions are, but given that, and I'll have to know their values that they observed, but given that, in theory, I can write out what this adjustment factor is. Okay, here's another more sort of perhaps more realistic example. I won't go into great detail, but suppose in my HIV resistance data, this is real data that I have. I had 633 samples and 91 features. And so here's a sort of a simple workflow that we might ask an undergraduate or a master's student in a regression class to do. Search for some important main effects in this data, and using marginal screening or some other method. Here I use a randomized marginal screening. That gives us some number of mutations. In the data I'll show you in a second, that was 23 main effects that seem important. And then the scientists realized maybe, oh, well, there's this important mutation out there called K65R that wasn't discovered in stage 1, but it really is important, so I want to include it. Okay, so we're going to insist that our model include this main effect. That's not a real problem. And then given those 23 plus 1, so those 24 main effects, I'll look for interactions. And the way I'll do that is I'll form what's called all the pairwise interactions and fit this into something like the lasso. And then at the end of that, I'm going to report p-values, point estimates, etc. Okay, so here's an example of the confidence intervals we might get in such a procedure. And I just use this example just because it's not really clear to me that, that the simultaneous inference or many other frameworks can allow us to do valid inference in this way, except if we collect confirmatory data, like a new sample, or perhaps we did something like data splitting, that is, held back some data in the first stage. So we can allow sort of kind of realistic data analysis schemes in this setup. And here's just a quick look at how much did I pay for selection in this context. We used the bootstrap to estimate variance to form the intervals that I didn't detail here. But here's just a comparison between the length of intervals if I had done the naive intervals and the conditional ones. And we haven't paid much of a price here, you'll see. We don't know a priori how much price we'll pay. If we were to do data splitting, then we'd expect this line to be 50% data splitting, this line to be about have slope root 2 roughly. So we didn't incur too much cost here. I have 2 other quick examples that I think I'll pass on. And I'll just talk about some challenges and then wrap up. So in practical concerns, there's a trade-off between the quality of the selection quality, that is how much information you can reveal with the queries, and power in the second stage, that is in the confirmatory analysis. And I don't have a natural way to trade this off. I don't really have a good sense about how to do that. There's some theoretical questions that are, of course, interesting. So based on what we know about a given model M, what can we say about selective models M star when we've arrived at them through this kind of this interactive result? Do we still have consistency of estimates? Do we have consistency in distribution in the sense of CLT? Rigorous results with high dimensional models are kind of hard to write. We don't have many results there, though these examples are practical in high dimensions. Computationally, we don't have theoretical results. So the computational problems are, how do I evaluate this zeta star? And we can do that with MCMC, but is my MCMC working? That's a question that's inevitably hard to answer. So, and with that, I'm just going to wrap up by saying that in order to address replicability, I think we need both this simultaneous and conditional inference. And I think I'll stop there.
We have questions? Yes. Someone is taking a microphone there, so the question can also be recorded.
Hi. From a statistical practice perspective, given that a lot of non-statisticians and mathematicians have trouble grasping the statistical methodology in a classical setting, have you had any experience with trying to convince non-statisticians to use this type of methodology to improve upon those problems we talked about to motivate this?
Of course I have. but that's a difficulty you confront when, even convincing statisticians they want to use it, who know the issues is another issue. So of course I've had issues. I think something that would hopefully be convincing are examples like this one, where you can show that if you just stick to the classical approach, you might be leaving things on the table. So you might be leaving power on the table. Then there's some motivation for, well, maybe I should think about how I can optimally use all of my data. But of course, anything in statistics, you'll have problems convincing anyone, in my opinion.
So we have time perhaps for one quick question. Let me just take the microphone.
So a follow-up on that, just like with the p-value, there's nothing really wrong with the p-value. It's how people use it, right? And the fact that there are millions and millions of people using it. So if we somehow succeed in convincing all of our collaborators to switch to say this paradigm or knockoffs or whatever the right thing to do is, can you anticipate any problems that will still arise just from the fact that there are millions of people using it and there are only so many data sets that they're looking at for a particular problem? Or will it just then go, say, for example, like cancer, breast cancer, that particular gene that they want to discover, there's still only so many data sets? Or will this really solve the problem if we could just convince them?
Well, so in order to use this, one has to be willing to describe explicitly how you arrived at these questions. So you should reveal what you've observed about the data. And so if people aren't following that, then this is not going to provide something. And on the second note about there's only finite data sets, well, if you really want to sort of follow this paradigm, I guess when someone publishes a paper, one should consider that we've conditioned on the results of this paper. And then there's less information left in the data. And reanalyzing data more and more, you'll get less and less out. And I think that's a reasonable property. It's not really attractive to scientists who have to pay for data if they want to reanalyze some data. But if you want to be rigorous about this, if you've read someone's paper that says this is an interesting effect to look at, and then you just analyze it, forgetting all the analysis that other paper had done, then you subject yourself to the same problems. So there's no easy answer. Yeah.
So we're out of time, so let's thank our speaker again.